Types of PCR. Recently, different variations of PCR have been developed that are based on the basic PCR methodology or reaction. And these modifications or variations have increased the performance as well as specificity of the reaction. और इसके अलावा जो modified forms हैं PCR की, इसको दूसरे जो मॉलिक्यूल हैं न्यूक्लिक एसिड के लाइक RNA, उसकी एम्प्लीफिकेशन के लिए भी यूज़ किया जा सकता है. So one of the variation that we are going to discuss in next topic, it is RTPCR, reverse transcriptase PCR. This RTPCR, it is used to amplify or to convert the RNA molecule, especially the messenger RNA, into DNA. So one can say that by using this technique, we can convert the messenger RNA into a type of DNA which is called as complementary or cDNA. And this is accomplished by using a special type of enzymes that are called as reverse transcriptases. It was discovered that certain viruses like the retroviruses that have the nucleic acid in the form of RNA, they can use the reverse transcriptase to convert its RNA into DNA during the replication process. So this ability of reverse transcriptases have been used to convert the RNA molecule into cDNA molecule. In conventional PCR, the enzyme that we are going to use, it is thermostable DNA polymerase, and it can only amplify the DNA template. So we can say that it will not amplify the RNA molecule. So because of this reason, reverse transcriptases, they have been used, because sometimes it is desirable to study different processes like the gene expression in different tissues, that how genes are transcribed and how they are expressed. And even in gene cloning procedure, sometime we require that it may be converted into DNA molecule, which is called as the cDNA. So for this purpose, we need DNA template that is formed from the messenger RNA by reverse transcription. So that we can provide the DNA polymerase with DNA template in the form of cDNA. So this process where we use reverse transcriptase enzyme to produce a copy of cDNA, it is called as reverse transcriptase PCR or simply RT-PCR. Now the question is that what may be the source of reverse transcriptases? Source may be a different type of viruses like avian myeloblastosis virus, or other viruses like the Meloni, or in other words, one can say that the leukemia virus. They may be the sources of reverse transcriptases. By using these enzymes, the first strand of the cDNA, it can be synthesized by using different strategies. कि हमारे पास मैसेंजर RNA है, रिवर्स ट्रांसक्रिप्टेज को यूज़ करते हुए हम जो फर्स्ट स्ट्रैंड है cDNA का वो कैसे सिंथेसाइज कर सकते हैं. So one of the method is that we can use random priming method, that we can use primer that can randomly bind with the target sequence of the messenger RNA. Another method is that we can use a short stretch of thymine residue as a primer. And the third method is that the sequence specific primer can be used for this purpose. So what may be the strategy? We are going to discuss in the next slide. And these strategies may be, first one, it is random priming. That is, random primer can be used for the first strand biosynthesis of the cDNA. And here these are different sites where primers they can bind randomly with the target messenger RNA. The second type, to use a short stretch of thymine residue or oligonucleotide. If you have the idea from our previous discussion that messenger RNA at the 3' end, it contains a short stretch of adenine residue that may be up to 200 adenine residue. And against these residues, a short stretch of oligonucleotide that composed of thymine, they can be constructed and can be used as a primer, so that the first strand biosynthesis is there. The third is that sequence specific primers can be used. So for this purpose, we must have the idea about the sequence of the messenger RNA. So these are the three main strategies that can be used for the synthesis of first strand of messenger RNA. So by using these strategies, now we can generate our cDNA. As I told you that the main step that is involved, it is by using a short stretch of thymine residue that can be used as a primer. It is the most common step that is used for the generation of cDNA. So the basic reaction we can explain with the help of another diagram, that how we can synthesize the cDNA. So first step, it is the first strand cDNA biosynthesis, and reverse transcriptase enzyme it is involved here, and this is the thymine primer that is complementary with the adenine residues. So ultimately it will lead to the synthesis of first strand of cDNA. Then in the second reaction, we can start the synthesis of the double-stranded DNA. So using cDNA product, we can use that, we can synthesize the double strand of the DNA molecule. Although first a few cycle they are biased. इस सेंस में जो बायस हैं कि ये बार बार फर्स्ट स्ट्रैंड ही सिंथेसाइज करेंगे, लेकिन एक दफ़ा डबल स्ट्रैंडेड डीएनए जनरेट हो जाए, उसकी कॉपीज आ जाएं, तो फिर इसकी जो एम्पलीफिकेशन है ये एक्सपोनेंशियल हो जाती है, एंड देन वी विल गेट द डबल स्ट्रैंडेड cDNA molecules.